thank you, Eric, and thank you for that introduction, and also thank you for inviting me to come back to Puerto Rico. I would like to say, as you heard, since 2008, I am the chair of a department. I am a reluctant chair, and I would like you to tell all your friends and enemies that there is a job going, okay? It needs somebody about half my age, or closer to half my age than my age, because I cannot handle this much longer. So I had to get that out to start off with. All right, so this also was a title given to me, and we will do what we can with it. Methods in statistical genetics, well, there have been, at least in human genetics, we first of all want to know if something we're interested in, there's familial aggregation, then we look for segregation, then familial co-segregation, otherwise known as linkage analysis, and then association. This has been the paradigm, very old one, which all of a sudden has disappeared. But nevertheless, I think some of it will come back. So first thing you have to decide is what kind of traits you're going to examine. I'm going to run through some of this fairly fast. Binary, ordinal, continuous, univariate, or multivariate. And typically, we've been looking at univariate, binary, and continuous. But these are things that have to be considered. Then, what kind of samples? Now, this, I think, bears much more consideration. You could have random cohorts of unrelated individuals, trios, mother, father, child trios, nuclear families, two parents and their offspring, or extended pedigrees. And now that we are interested in common diseases, there's often a lot to be said for looking at random cohorts. I do know of studies. I mean, if you're interested in hypertension, if 20 or 30% of the population have it, there's no reason why you shouldn't. And I know of one study which is looking at random, well, let us say pseudo-random as you get, a random sample of large extended pedigrees throughout Europe. And so I think we need to consider that. Otherwise, we have selected cohorts. And the selected ones could be case control, which is now seems to be all the rage. Trios again. In other words, we might select parents and offspring because the offspring is affected. Our nuclear families in which at least one child is affected, or extended pedigrees where somebody is affected. And all these things really need careful consideration. And I don't think it's at all obvious, as you'll see as I go on, what we should be doing. Even with the next generation sequencing, which I think is going to take place and have a lot of, and be very, very important, and it's going to maybe get rid of microarrays, I don't know. But certainly we're not, for a long time, I think, going to be able to dispense with family data. And you'll see that as we go on. Okay, so what are we looking for? And, you know, typically people are not always sure what they're looking for, and I think that needs to be decided first. Are you looking at the marginal effects of one locus genotypes, or the marginal effects of alleles or haplotypes? Or are you looking for the joint effects of various one locus genotypes across the whole genome, the joint effects of alleles or haplotypes, the joint effects with environmental factors? Now, the buzzword these days is gene by gene interaction, gene by environment interaction. Those are buzzwords. If I had my way, we wouldn't be looking for them. We'd be trying to get rid of them if we could. And sometimes we can. But we just certainly, and so I'd much prefer that we talk about the joint effects. We're always interested in the joint effects. We may or may not be interested in interaction in the statistical sense. I think we're interested in the biological sense, but that is not necessarily a one-to-one correspondence between the two. Now, before you start looking at genetics of a trait, it wouldn't be a bad idea to know that it's familial. It doesn't really make much sense to me to look for the genetics underlying a trait if it's not even familial. Now, one of the things people do is they say, well, let's look at the heritability. Fair enough. There's lots of problems with this. There's different definitions. There's the broad definition of heritability is the proportion of variability due to all genetic differences in the population. Narrow is the proportion of variability due to additive genetic differences, because that is what's transmitted from parent to offspring. But I want to point out that heritability is a population-specific concept. I don't know why, but people will tell you the heritability of this trait, that trait. They'll never tell you what population. It's as stupid to talk about the heritability of a trait X as it is to talk about the heritability in a population Y. The two go together. There's no reason why the heritability of a trait should be the same in all populations, and in fact probably isn't. But people talk as though it is. Makes no sense to me. So, now if you look at the remaining proportion, people say, well, it's environmental or measurement error. But that's only true if there's no what we call gene by environment interaction in the statistical sense. So therefore, we would like to minimize that interaction from a statistical point of view. We're always interested in joint actions, but we don't want statistical interaction if we can get rid of it. And that's going to be a major point as far as I'm concerned in any statistical analysis. All right. Now, how do we estimate heritability? I mean, I've said, people are doing this because they say, well, you've told us we have to show the trait's familial, so now we'll go and estimate heritability before we start. Fine. So, all estimates in humans make assumptions about phenotypic correlations that are due to the common environment between two relatives. All right? You've either got to assume it's not there, or that it's the same for different kinds of relatives. And these environmental correlations would, if you don't do something about them, would be expected to inflate an estimate of heritability. Next thing is, people talk about the heritability of schizophrenia, for example, heritability of Alzheimer's disease. And typically, when they say that, they're not talking about the heritability of that trait at all. What they're talking about is the heritability for some latent liability to the trait. All right? Now, I will tell you, if you assume, for example, that schizophrenia happens to be due to one single gene segregating, you get a heritability of 15%. On the other hand, if you assume it's polygenic, and they've got a nice normal distribution for this liability, the heritability is about 75%. So it is rather stupid talking about the heritability of a binary trait unless people tell you how they got it. All right? When it can vary from anywhere from 15% to 75%, one wonders if they know what they're talking about. All right? So the assumptions you make about the distribution of this liability are critical to the estimate you get of heritability. All right? And by the way, if you have any questions as we go along, please feel free to ask questions. All right, segregation. And, people tell me, oh, segregation, that's passé now. I disagree. I disagree completely. Perhaps we need it more than ever. So, you may have unaffected or two distributions determined by a dominant or recessive gene, or you may have three distributions. Well, now, what do you think we're doing now when we're calling these genotypes with microarrays? That's what we're doing. we've got these distributions. All right? And in fact, some methods have recently been suggested for using mixtures of distributions in order to call them, which is correct, which we were doing years ago. And but what we were asking when we're doing segregation analysis is, can the dichotomy or trichotomy be explained by Mendelian segregation? That we use family data for. But if we don't have family data, we can just ask the question, can we separate out the distributions? Which is really what the raw data are before they come to you after somebody's messed them up and come out with some output. All right? They're really continuous distributions. All right. Now, segregation analysis. People don't do it anymore. I think that's wrong, by the way, which I've just found out very recently. It's a big difference whether you use discrete traits or continuous traits. I was very surprised. I was just trying to show that something was familial, and it turned out that it fitted Mendelian segregation. I was very surprised. And now some people say, we used to know this. We knew so much. Further back I go, the more we knew. When I started, we knew we had 48 chromosomes, and all of a sudden it changed to 46. These things we knew. We knew years ago that discrete traits were Mendelian and continuous traits were not. Well, that's completely wrong. You can have discrete traits that are not Mendelian, and you can have continuous traits that are Mendelian. And so, but it makes a big difference, the analysis which you have. Now, you may have multiple alleles and multiple loci. This, for segregation analysis, is a problem and not a problem. Multiple alleles is a real problem, all right? When you've got lots of different alleles and therefore lots of genotypes, you've got a mixture of lots and lots of distributions, we can't handle it. Multiple loci, not necessary. If you think of all the monogenic diseases that we found by segregation analysis, not one of them, as far as I know, not one of them was the disease due to a single allele. There are lots of disease alleles, yet segregation analysis could find them. All right? Because it assumed they all had the same effect, and that worked. And it still works sometimes. So you really have to realize what's going on. And then you have to ask yourself, what models are realistic? People are doing analyses now which, and I think, are completely unrealistic from a genetic point of view. But they say, well, we have to look at everything because we don't know what's going on. Well, yes and no, but does it make sense to look over here where the light is, where what you might want is over there, where it's dark? Doesn't make much sense to me. So you really have to, I think, in any kind of analysis, segregation analysis particularly, and other analysis, you have to ask yourself, what makes sense? The problem of ascertainment is too often ignored. It was very easy when we did segregation analysis with SIB ships because we had a method of allowing for it. So segregating SIB ships, what happens is that the proportion of SIBs affected is larger than expected on the base of Mendelian inheritance. Even if you do have Mendelian inheritance, if you select the SIB ships because they got the trait, clearly you're going to get overestimate. And so you have a likelihood, and you condition on the mode of ascertainment, and that's fine. And as people have did that a long time ago. Now, what about large pedigrees? Some people say you can't do it, it's impossible. Well, it is, and this is about the only equation I'm going to have anywhere in the whole slides. It's, in theory, solved, but what you need to know is what I call the proband sampling frame. And when I first used this word back in 1979, I thought I'd use English that people would understand, and I said, but when I use this phrase, I mean this. And of course, people never read that, and they thought I was absolutely stupid. So what do I mean by the proband sampling frame? You have a pedigree, all right? And the people who are in that pedigree, they are there either because they have some trait, some extreme value, say, all right? But forget that. They're in the proband sampling frame if, irrespective of what their trait is, they could be in your sample and bring your pedigree into the sample. And if they weren't there, then they're not in the program sample. So being in the proband sampling frame has nothing to do with whether you're affected or not. It's just being possibly, if you were affected, could you come in? All right. And when you, I'll show you in a minute how you could do it. Then there's this other approach called the mod score approach, which is very interesting, which in a nutshell said, when you're doing linkage analysis, if you maximize the LOD score, and we'll talk about that later, if you maximize the LOD score, you don't have to worry. It takes care of everything. It was rather intriguing. Nobody said it was efficient. It would just give you valid estimates of what you wanted. I'm not going to go further in that. We can talk about that later on if you want. But what we were trying to do, of course, was we're trying to, you've got to decide, are you trying to find genes or are you trying to evaluate their effects? If all you're trying to do is finding genes, perhaps ascertainment is completely unnecessary. Forget, I mean, unnecessary to allow for ascertainment. You don't care, because under the null hypothesis, there's nothing there, right? So under the null hypothesis, you can go ahead and do all your methods ignoring ascertainment. But if you're trying to evaluate their effects, then you must allow for ascertainment in the analysis. And if I write it very simply, if for a particular pedigree, and I'm going to say we've got the whole pedigree, PR, we define the subset of individuals who are in the proband sampling frame, that is, could have caused your family to be in the study, sampled, regardless now of whether it's affected or not, and we let those be the PSF. So if we maximize the likelihood, this one that says it's the likelihood for the whole pedigree minus the likelihood for those in the pedigree sampling frame, which is the same as if you this is log likelihood, so that's like taking the ratio you've divided by the probability in the proband sampling frame, that's conditioning on that, then you get consistent estimation of all parameters. Not necessarily efficient, but you'll get consistent estimates. So remember, if you're only trying to find genes, this is completely unnecessary. But if you want to evaluate the effects, this becomes very necessary. And also, of course, there's problems with this approach if you have case-controlled data, because you selected these case-controlled data. They're all there, and you're going to throw out all that information. You're going to condition it out, so it won't work at all. All right? So when you're doing case-controlled data, if you want to evaluate effects as opposed to find genes, that's extremely difficult. All right? Extremely difficult. Can be done. All right? But not, and you will, well, I'll say always, virtually always, it's impossible unless you know the prevalence of the disease in your population. That's not perfectly true, because if you take your controls to be people who are, to be a random sample from the population, as opposed to those who do not have the disease, then you have some information on prevalence. All right? So that's a way around it. Otherwise, you get odds ratios, and the only way you can go from odds ratios to what you want is by knowing the prevalence of the disease. So those of you who are only interested in finding genes, you can forget a lot of this. All right? I maintain we have other interests in life. All right. So, linkage analysis. And basically, linkage analysis just finds locations. That was the idea. Very simple, and then, of course, you tend to have a model. Now, there's a statistical model, which has to be correct, but there's also the genetic model. Okay? You think there's a gene here, you think there's segregation here, and you model everything, all the penetrances, the allele frequencies, and all that. So if you have the right model, clearly it's the best thing possible, okay? And for complex diseases, the model is usually never correct. So but now the model-based analysis is often validity robust. In other words, by that what I mean is you get the right kind of answer in terms of not getting, you control your type 1 error. We'll come back to that. All right? That doesn't mean to say you get the right answer. You're going to control your type 1 error. All right? Now, some model-free methods are validity robust for heterogeneous diseases. Others can have increased type 1 error, so you've got to worry about increased type 1 error. I'm going to go through some of these statistical things later on. This is just an overview for those of you who know anything about linkage analysis. And so the other thing is in model-based linkage analysis, you incorporate heterogeneity by assuming a simple segregation model, but at any genomic location, you assume some families are linked, some are not. This leads to this wonderful thing the statistic people call HLOD, okay? And people will often quote this thing HLOD and not even tell you what it means. And people will quote LOD and not even know what it means. We're going to come back to that later. H is the heterogeneity bit. LOD, people think, are the logarithm of the odds. That's what they think. And this is certainly not true in linkage analysis. All right. So, and then people do what is sometimes called joint segregation and linkage analysis. And whenever you are doing something where you're estimating parameters of the segregation model, you must allow for ascertainment. And there are many, well, let's say I remember several, just to be careful. It's situations where papers have been published in very good journals, even in one case won an award, did not allow for ascertainment. The result was rubbish. Just want to let you know. One thing you do not do things because everybody else does it. That's hardly a good reason. All right? You need to think. All right. Now, multi-point versus two-point linkage. Those of you who've done any linkage analysis at all. Multi-point is more powerful in principle, but it's very sensitive to misspecification of the assumed marker genetic locations. We assume we know where they are. If you get them mixed up, you get a mixed-up answer. And, of course, it's only relatively recently we know that on chromosome 8, I believe, in Africans, there's an inversion of a whole chunk. So people had all these things the wrong way round. So, that was giving us lots of mistaken answers. Now, people said, well, because of that, we should start with two-point. Okay, just look at the linkage of your trait with one location at a time, one locus. But if you have SNPs, and that's all you have, single nucleotide polymorphisms, which we assume are only diallelic, okay, then you lose a lot of power. And I assume, but everybody does assume they're diallelic. AFI even says, well, we have an unbiased, well, they go things unbiased over the whole genome. No. They only look at the diallelic ones because they can't call the triallelic and quadriallelic ones. All right? Everybody thinks SNPs are diallelic. I've been told by somebody who's looked for them, about 30% of them are triallelic. There's no reason why they should be diallelic, except that people can't call them, so they throw them out. So, there's all these things that people don't think about, which really worries me. So, the other thing is that most multi-point algorithms assume all markers are in linkage equilibrium. In other words, they're independent. Actually, one of them, it can be shown, doesn't. And you can have very, very close SNPs and do a model-free linkage analysis, and it doesn't matter. Shown in a paper, and if you're interested, it was one of my students, Xing, X-I-N-G et al., and I'm trying to think I think it was in Human Heredity. It got published like a year or two ago. Okay. So, what is this model-free linkage analysis I'm talking about? Well, it takes pairs of relatives. It's very simple, really. We correlate relative pair similarity or dissimilarity for the trait of interest with relative pair similarity or dissimilarity for a marker. We're looking at similarity of the trait between relatives, similarity between the marker for the relatives, and we're correlating these two similarities. That's all there is to it. Okay? And if there is linkage between a trait, some trait locus, the marker locus, you get a positive correlation. Very simple in concept. Of course, the way we do the analysis might be much more difficult. Now, some people say, aha, we'll just look at affected relative pairs. And we ask the question, do affected relative pairs share more marker alleles than expected if there's no linkage? Fine. Except now, you have no controls. From an epidemiological point of view, you should be comparing this with the discordant relative pairs. People never do. Does it cause problems? Well, maybe it has, probably rare, but logically, we have to compare. Now, what we're doing when we do affected relative pair analysis, we are assuming that our markers are behaving beautifully. They're Mendelian, there's no segregation distortion or anything like that. So it's a shortcut. Right? Remember that, it's a shortcut. Just by the way, as heritability, Sir Ronald Fisher described it as a shortcut. All right? And we've got to always beware of shortcuts. Now, this is what everybody's into now. Association. All right? It's the end of the world. All right? Well, we thought it was maybe, I don't know, 5 or 10 years ago, and all of a sudden, yeah, it's produced something. What has it produced? We're not too sure. So, what are the causes of association between a marker and disease? Well, it could be chance. That's why we do things like Bonferroni correction, all that. It could be stratification, population heterogeneity, and we've looked for that. It could be very close linkage, and hey, we hit the jackpot if it's pleiotropy. The allele that we've said is associated is the causal one. That's what we really want. Okay. So why do you perform this association study? And if you don't know why you're performing it, you shouldn't perhaps perform it. Maybe you want to find the genomic location of causal variants. That's one possible reason. Another is to estimate the attributable risk due to the causal variants. That's a completely different reason. Or you want to predict clinical outcomes from the base association variants, get better prediction, better treatment. That also is a different. I mean, if all you want is to estimate attributable risk and predict clinical outcomes, you really don't care about the biology behind it, as long as it works. Right? If you want the genomic location, then you don't care about your estimates of your effects. Right? So we need to be very clear what we're looking for. Now, this association versus linkage. I think a lot of people have just made up their minds. But it's not so clear to me. See, association is at the population, linkage is intrafamilial association. So you need families. Association pinpoints alleles, the actual variants. Linkage pinpoints loci, the location. It's not the same thing. And then they always said, well, association is more powerful. That's why we should do it. That's why we started doing it. It's more powerful. Linkage is less powerful. Clearly. Oh, but now with allelic association, we require more tests. Linkage, fewer tests. So, hey, is this powerful thing really more powerful? Think about it. If you're doing more tests, you've got to worry. You need a much lower significance level. So what you gain on the swings, you've lost on the roundabouts. Association is much more sensitive to mistyping. You can't do much about it. Whereas in the case of linkage is less, and you can also check. You can check the typing to see if you've got Mendelian segregation. Allelic association is very sensitive to population stratification. Linkage is not. So which do you do? That's a good question. Well, I'm not going to tell you which you should do, but I think it is stupid to give up linkage. That's my own belief. I'm not saying you should only do linkage. I'm just thinking it's stupid to give it up. The point is, you've got advantages. Often the DNA is already there. You collect it long ago. All right? Haplotypes are more easily determined. You can use linkage information to increase power. You can use linkage to protect against spurious associations. And it may be essential if there's a lot of allelic heterogeneity. One of the problems we have with these association tests is a lot of allelic heterogeneity, which we know from monogenic diseases probably exists, so we don't know what's going on so well. Okay, disadvantages, you've got to get family members. Do you need a larger sample size? I don't know. And certainly it's very poor for estimating attributable risk or linkage. Okay? So, if you look at samples without linkage information, well, should you pool your samples or should you not pool them? Now, people have, I think in the latest GWAS things, they said, oh, well, this is so cheap, we won't even bother pooling. Now, when it comes to next generation sequencing, or at least when it comes to some of what they're doing, they're pooling things. Again, we're going back. So we always seem to go backwards when we've gone too far forwards. There's a reduced typing costs. Disadvantage, you've got to prepare your pools, you get less haplotype information, you've got to worry about the accuracy of the different measures and the calling. So pooling has advantages and disadvantages. Now, what is the information from association? You can have trends or differences in allele frequencies. That's what just about everybody looks at. They forget everything else. You can have trends or differences in the LD structure. The population geneticists are looking at this. You can have trends or differences in Hardy-Weinberg disequilibrium, which people seem to think doesn't exist. All right? And they use that as a QC. Don't have Hardy-Weinberg equilibrium? Throw it out. That's throwing out the baby with the bathwater sometimes. Okay, so this is a nice diagram, very quick diagram. You can see. So this is what people have been doing. If they look at the allele frequency difference, they use what's called the Cochrane Armitage test. If they look at this one here, they look at the contrast between the Hardy-Weinberg disequilibrium cases and controls. Here you look at the LD contrast here, down here, and then you can put two together, D, that's here. This is, when you put in these together, you look at the genotype frequency different tests. SNPs, that gives you 2 degrees of freedom. Here you've got the haplotype-based test, but assuming Hardy-Weinberg equilibrium. Nobody's ever suggested doing this, probably for good reason. It's probably useless. But this here is the phase-known haplotype, the genotype test. What I do want you to notice here, though, is that if you decide that what is true is the null hypothesis for either of these three tests, it doesn't imply there's no association. It just could be you looked at the wrong one. On the other hand, if you say you are sure you have the alternative for any of them, then you have association. Okay? This is the basic idea I want to get across. So, joint effects of multiple factors. So, on the statistical point of view, they may be additive on some scale, in which case, find that scale, or they necessarily require interactions. Now, I'm not saying that we certainly want to look for biological interactions, but for genetic purposes, statistical analysis, we want to avoid interactions as much as possible to get more power. And maybe some people suggested, well, you look at the main effects first, and then if you find something there, then you look at interactions. Don't think that's a very good idea myself. But the point is, if you have parsimony, you have more power. So if you have a model where you can say, on this model there's very little interaction, statistical interaction, you'll get more power to find those biological interactions you're interested in. Okay. Association analysis, as I've said, you can have unrelated cases and controls. It will detect causative loci. It will detect linkage disequilibrium. It will also detect heterogeneity stratification. Right? So you've got to be careful. But if they're matched on ethnicity, then that helps. You could have relatives, the pseudo-SIBs, in other words, the SIBs that never were. That's a bit like the TDT test is using that. It's comparing what you have in your affected SIBs with the SIBs that never were, okay? Or other relatives, okay? You can use genomic control in the sense of estimating your ethnicity with other markers. But genomic control is something all by itself. Very simple idea, and it's mainly used when you're doing these case control studies and you're worried about stratification and what they call cryptic relationships. You calculate an association statistic for your candidate locus. You calculate the same association statistic from the same sample for a set of unlinked loci or unassociated loci. I don't know how you know that, but you assume that maybe, well, you do it for all loci that you have on the assumption that most of them have very little effect anyway. Okay? And then you determine the significance by reference to the results for the unlinked or unassociated ones. All right? So this is pretty good, so long as, and even you can't get, if you do it by reference to all of them, that's conservative. You use permutation tests and what have you. All right. Now, I want to talk a little bit about statistical inference, very quickly. All right? This is general. I don't think enough difference has been made between significance testing and hypothesis testing. And then, of course, decision-making is where we should be. Jack Kiefer, unfortunately, died very young. I had the good fortune to take a course for him before he died. And that's what we really want to do, is to make decisions with multiple hypotheses. Okay. Significance testing, I think you all know, you determine the null hypothesis you tested, such as no linkage or no association. You develop a criterion, something you calculate from the sample, whose distribution you know if the null hypothesis is true, and then you determine the significance level, that's the probability of the observed sample or anything more extreme if the null hypothesis is not true. That's the end. You end up with a p-value. That's significance testing, all right? So, this is Fisher's own words, though I can't find them again. I keep trying to find, I know I read this from a paper of Fisher. A more extreme sample is any sample that, even more than the observed sample, would alert the researcher to the possibility that the null hypothesis is not true. Okay. So you get a p-value, it's a one-sided, two-sided, possibly multi-sided, that's terrible, and you get a logical disjunction. Very simple. Either the null hypothesis is not true, or you observed a rare event, the rarity of which is reflected in the p-value. That's the end. Then, of course, we have the people who come and do hypothesis tests. Due to people like Neyman and Pearson, you do the same thing as before. You determine the null hypothesis. You develop a criterion. You know its null distribution. Then you say, aha! I'm going to define all the possible outcomes. Some, if you fall there, I'm going to reject. If you fall here, I'm not. And you're going to control this type 1 error to alpha. This is the way it goes. If the criterion falls in the rejection region, you reject the null hypothesis in favor of the alternate. If it does not, you accept. If it falls, well, you take it in one or the other. So, typically, you know, you write a paper, and I'm not thinking now in terms, I'm thinking of maybe before we did all this multiple testing, 0.05. If it was greater than 0.05, you said not significant. It's less than 0.05, you said significant. So I once submitted a paper, and I said the significance level was 0.06, and one reviewer said, well, it's not significant. Huh? Huh? Even Fisher didn't say you had to have 0.05. Nobody said you had to. It was just something for convenience in the old days because you had to calculate these things. 0.06, it seems to me, that's pretty close to 0.05. Who's to say it's any worse than, much worse than 0.04? Except the editor. He wants a decision rule. See, that's the point. So, this is the general idea in statistics. You have a model. Model's assumed to be true. There's a null hypothesis, and there's an alternate. And we have these two types of error. Probability of rejecting the null hypothesis when it's true, we call that alpha, type 1 error. Accepting the accept null hypothesis, reject is the alpha, and accept is beta, and we can have a little bit, I won't worry with that. So types of error. Type 1 error, rejection of the null hypothesis when it's true. Type 2 error, acceptance of the null hypothesis when the alternate is true. And I'm going to give you type 3 error, which we used to talk about in the old days, and it's been brushed under the table. Huh? The model's wrong. If your model's wrong, all bets are off. But, you know, but people don't even look at what they do in another system. They don't call it type 3 error. Okay. So, we have a simple dichotomy. It's probably you learned the true state of nature, the decision made. You have the number of counts A, B, C, and D in each cell. And the type 1 area alpha is C divided by A plus C. And the type 2 area is beta is the other way around. I'm pretty sure this is all. If it's not old hat to you, then you probably don't want to hear it anyway. But what I do want to get on before we get too far. Let's see. You need to, the probability, what you're really interested in. This is what you're really interested in. You tell the researcher, hey, this is the probability of the error if the null hypothesis is true. Well, I don't know if it's true or not. That's what I'm trying to find out. No sensible researcher has the least interest in that. What the researcher wants to know is, what's the probability of error if I make the wrong decision? And that's what's called the posterior probability of type. Posterior probability of type 1 error. And it depends on the prior probabilities, which we don't know. That's the problem. But is it a problem? We'll come to that later. Odds. Odds is very simple. The odds of an event or hypothesis, the probability of the event, and the hypothesis, the event or the hypothesis given the data. And the odds is 1 minus that. Very simple. Very simple. You have two possible events, two possible hypotheses. It's the probability of one given the data divided by the probability of the other. Okay? So if you have some parameter, it's either theta 1 or theta 2. This came out very funny. It should be that Q1. I don't know how that got there. That should be theta 1 or theta 0 in there. Somehow it got transliterated in whatever program they have here. I think it's correct in your handout. So Barnard called this the forward odds. That's what everybody thinks of as odds. Okay. Then you've got the likelihood. Well, likelihood is completely different. Okay. So it's what it is proportional to the probability of the data given the hypothesis. Now, if you have two simple hypotheses like this, then the likelihood that theta is 1 versus theta 0, it's interesting that Q came out there. Don't know how. Is the probability that is the data given that the other way around. So the point is that the likelihood ratio is what Barnard called the backward odds, all right, as opposed to the forward odds. So odds, now, Newton-Morton, you see, you can either get the forward odds, the odds of theta 1 versus theta 0, or the backward odds, all right, and by the way, Barnard used natural logarithms. The geneticists don't know how to do that. They can only take logs to base 10. All right. So Morton, you see, brought this in. And he said, well, we're going to take this, you see, and this is what is known as the Lod score. And I see you've got a Q. Well, I think it's right in your handout. And it is not the logarithm of the odds for linkage. Never is, never was, never will be. It's basically, it is a likelihood ratio. And it's completely different. And if you don't understand that, we can talk about it. Okay, multiple hypotheses. Well, first of all you're really interested in is the false discovery rate, the probability of error in stating the alternative is true, or the true discovery rate, which is 1 minus that, and in your handout, there's a knot in here. Throw that knot out. I don't know how it got there. Just cross it off, okay? So those are the only things you're interested in. Why would you be interested in anything else? Well, and the nice thing is that when you're doing all these hypotheses, you can control those, you can get them without assuming prior probabilities. This is the famous Benjaminian Hochberg paper, The Statisticians, 1995. It was not put this way. It was talked about multiple comparisons. To me, the nice thing is it says, hey, you don't need to know any priors. Isn't that nice? Exactly what everybody wanted. Okay? But it's pretty useless if you never state the alternative is true. So you still have to look at power. All right? It's pretty useless if you never. This is saying probably when you state H1 is true or 1 minus that. That's all you're interested in. But if you never state anything, it's not going to help you. So you've got to have power. All right. Validity. Test is valid. Basically, it can be valid if the type 1 error is okay. And sometimes it's okay and sometimes not. People tend to look at the 0.05 level, and when they get the 0.001 level, you know, some of those things you see now. Oh, you see these p-values of 10 to the minus 20, 10 to the minus 40. If you believe those, you are absolutely crazy. All right? They're all based on assumptions which don't hold. For God's sakes, what is it, 10 to the, once you get to 10 to the 18th, that's more than the number of particles in the universe or something? I don't know. You're crazy if you believe those things. I know people put them, maybe I probably do too, but I'm crazy too, all right? But that's what people want to see, all right? So you should either get the correct value for a finite sample or use some simulation or permutation technique. You've got to have power, okay? A test that correctly uses more of the information available is more powerful. But if you misuse that information, it won't be more powerful. You know, I always used to think more data, better. Not if you misuse it. And there's a lot of misuse of data. All right? You use it in your statistical test in the wrong sort of way. Model-based analysis, there's a correct model, it's always better. Okay. Otherwise, you may be in terrible straits. Robustness, that means it's pretty good even if your assumptions aren't right. Just go on with this. Okay. And in genetics, typically, most genetic analyses, these are critical assumptions that if you're doing anything that requires relationships, known without error. Anything requires marker allele frequencies, they're known without error. And, of course, the marker typing data is correct, or else you allow for an error in your likelihood. Okay, summary. My summary is that purpose is paramount. All right? Next one is power is important at the design stage. And I fully agree with Terry. Probably impossible to get at, but still important. All right? And that's one of the things I'm thinking about how you can do. The false discovery rate and its complement the true discovery are they're the only things that are important at the analysis stage. Who cares about anything else? All right? And beware of type 3 error. It's much more prevalent than you think. All right? And beware of results that depend on asymptotic null distributions. These p-values are 10 to the minus 20. They're just rubbish. Okay? So, distinguish between evidence and belief. A lot of what geneticists do is based on belief, dogma, not evidence. I can talk more about that if you want. This, I think, is the important thing. I have this outside my office, the Rosetta Stone. I stole it from the British Museum, by the way. And I think we're getting closer to the Rosetta Stone, and the next generation sequence will certainly help a lot. So I always end up with this last slide. The thing is, you know, people in their last, they put up all the people who collaborate, put all the people in their lab and picture of that. I give credit where credit is due, the National Center for Research Resources. They've been funding me well for over 20 years. I hope it'll go on for another 20 years. And what do I do for them? I have this thing called the Human Genetic Analysis Resource. You look, see what it does there. And by the way, I was asked to bring some brochures, to send some brochures. I did. Is Marivalisa there? I don't know where they are, but they're somewhere at this institution. Don't ask me where. If you want, interested, and we produce this statistical package. Okay, so I will leave it at there to give at least a few minutes for questions.